Hello students, this is my part 7 video of angiospermic reproduction. In this class we can discuss the development of embryo, that is dicot embryo. So this development of dicot embryo or embryo is also called as embryogenesis. Embryogenesis in dicot, and the branch of science deals with the study of development of embryo is called embryology. So this development of embryo in dicotyledon plants was first discovered by Hansten in Capsella plants. So in the last video we have already discussed about the double fertilization and triple fusion and how the zygote is formed. So the development starts from the zygote to the matured embryo. So first of all we have to discuss what are the steps, and then finally we will discuss in detail in diagrams. So first, that is zygote, then next stage is oospore, next the division is there, that is called two-celled stage. Then next step is T-shaped embryo. Then quadrant stage. Then octant stage, then globular stage, then heart-shaped embryo, then finally reaches the mature embryo. So in this way, from the zygote, then oospore, then two-cell stage by the mitotic division, then T-shaped embryo, then quadrant stage, then octant stage, and then globular stage, then heart-shaped embryo, and finally the mature embryo. So we will discuss in detail diagrammatically how this occurs. So the first stage is zygote. So we can draw the structure like this, and we have already discussed this zygote is 2n and the product of fusion of one male gamete with the ovum. So this zygote in the next stage, it will absorb nutrients, absorbs nutrients, increase in size, and the main source of nutrient is nothing but the endosperm, and gradually it increases in size. In this stage we can name it as oospore. So then the first division occurs, the first mitotic division, occurs and gives rise to two cell. So this upper cell, particularly here, this is called red in color apical or terminal cell, which faces towards the chalazal end, This is the orientation of the two cells after the division, and this lower one is called, black in color, is basal cell, or we can tell suspensor cell, because it will later on produce the suspensor. Suspensor helps the embryo to enter into the endosperm and absorb the nutrients. So in next, and this is the orientation, is the basal cell faces towards micropyle end. So we can see also how inside the ovule this takes place. If we draw the structure of ovule, what we have already discussed in the previous videos. So here the embryo sac, and this is the micropylar end, and this is the chalazal end. So this two cell stage, first form, is like this, and here this implies your apical, red one is apical, which faces towards the chalazal end, and the basal cell faces towards the micropylar end. So next we will discuss, this will divide, these two cell stage then divides. So the lower one, or the basal cell, divides in transverse division. So we can write this is basal cell, but the apical cell divides, that is your longitudinal divisions, producing two cells. These two are called apical cell. So it is clear here that the lower one, basal cells, divides transversely, and here the apical cell divides longitudinally. So we can write here longitudinally, and basal cells divide transversely. So here it appears like the T shaped, so this is, we can write T shaped embryo. This is T shaped embryo. Then later on again the development occurs, and it will, particularly the apical cells divides. This is the basal cells, and this apical cell divide transversely, so produces this clear demarcation by the red color and black color. So in this 4 number of cells are produced. So for that, this stage is named as quadrant stage. Quadrant stage. This is called quadrant stage. Then this quadrant stage means, quadrant means 4 numbers of cells are produced. So later on in the next stage, what happens? This basal cells divides further. The basal cells divides further. Basal cells, this remains as such, and the basal cells divides further to form, that is, to forms 4 or we can tell 6 to 10 numbers, 6 to 10 numbers of, that is, suspensor. And by the increase in this suspensor, what happens? Gradually the embryonal cells, particularly what we call the apical cells, later on gives rise to the embryo. It will push the embryo into the endosperm. So we can write here suspensor. This is suspensor, and this base cell we can write hypophysis. Hypophysis. And this hypophysis later on produce the root cap. In future it will produce the root cap, and this base cell is also called hostorial cell. Hostorial cell, it absorbs food. It absorbs food. It absorbs food. So this is the stage where this is also the nothing but the this development occurs. This is followed by the this quadrant stage, and later on these will divide to form, particularly these apical cells, this will divide to forms the octant stage. So the octant stage, by the division of these apical cells, in this way you can draw the structure. By division they will form 8 number of cells. And we can here draw the, that is, suspensor. So this is called octant stage, and these cells, particularly these cells, upper one, are called as epibasal cells. Epibasal cells forms the cotyledon, forms the cotyledon and plumule. Plumule, you know that plumule gives the shoot system. So in this octant stage we are giving emphasis that the 4 embryonal cells really divides. The 4 embryonal cell divides to form octant. Next stage is called, that is globular stage. Globular stage in this case, this epibasal cell and particularly the embryonal cells, what we can tell also, it will divide in both directions and produces a mass of cells. Mass of cells are produced. And globose structure appears, and this is also called as pro-embryo. This is also called as pro-embryo. And here the suspensor is there. So this is called your globular stage. Particularly due to the division, the octant divides periclinally. We can specify here also the octant, the previous stage divide periclinally. Peri means to surround, clinally means this lateral direction, to form this pro-embryo. Then the next step is called your heart-shaped embryo. Heart-shaped embryo, we can see the structure. So heart-shaped structure is found. Heart-shaped embryo, 2 lobes are very distinct, and these two lobes are very distinct, Lateron gives rise to, which gives rise to the cotyledon, and this anterior portion, this depression portion, which is called as the plumule, which will later on produce the shoot axis. So next, this heart-shaped embryo finally gives rise to the mature embryo. So the mature embryo, we can draw the structure, that is your structure like this. structure like this, and this middle portion is called embryonal axis. Embryonal axis, or also called tazellum. And these two are called two cotyledons, and this region is called plumule. This is called plumule, and this lower portion is called hypocotyle, which lies just below the cotyledon when the embryo will germinate, and this gives rise to the radicle, which gives rise to the radicle in later on gives rise to the root system, and this posterior part forms the root cap, and here you will find this is called hostorial cell, which absorbs the nutrition, and this is called suspensor, and this is the structure of mature embryo. So inside the seed we will find this mature embryo, or sometimes also called torpedo shaped embryo is also called. So in this way, inside the seed, or we can tell the ovule, the complete this dicotyledons embryo is prominent. So very important thing is this presence of two cotyledon, this plumule, which will later on gives rise to the shoot system, radicle gives rise to the root system, where this act as the root cap region, and this is hypocotyle region when later on germination occurs below the cotyledon, hypocotyle, and above one is called the epicotyle. So this is all about our development of embryo, that is dicoty, particularly in the dicotyledonous plants, and this embryology plays very vital role, and the father of, according to our data, or we can tell the very importance of the some data, like your father of embryology is C.F. Wolf, and father of Indian embryology Professor Panchanan Maheshwari. He has significant contribution towards the embryological development of plants, and he wrote a book that is an introduction to embryology of angiospermic plants. He wrote a book, an introduction to embryology of angiospermic plants. This is all about dicotyledon embryo development. In next video, I will discuss the monocot embryo development.